In this talk, we will learn about homeostasis and the role of hormones in maintaining homeostasis in the animal body. Homeostasis is the ability of the animal to maintain a balanced and stable internal environment within the body. Different animals have evolved different physiological processes to control their internal environments and maintain homeostasis. This includes maintaining an appropriate solute concentration in the body and body fluids, water levels in the body, as well as maintaining a steady internal body temperature, especially in the case of endothermic animals like birds and mammals. Different animals have different internal environment conditions, and hence their homeostatic conditions can be different. For example, the optimal body temperature of endotherms like humans are usually around 37 degrees Celsius, but birds, which are also endotherms, have an internal body temperature that is slightly higher than humans. When we look at animals that are not endotherms, like turtles, which are reptiles, we can observe that their body is able to maintain a higher range of temperature, for example, from 25 degrees Celsius to 35 degrees Celsius. The water needs of different animals are also different, as well as the solute concentrations. Maintaining homeostasis is critical for the functioning of the animal body because homeostasis allows the cells that make up the animal body to function efficiently. It also allows them to communicate with one another efficiently. Homeostasis allows the body to sense changes that deviate from the ideal conditions. It also helps the body to respond to bring back the conditions to what they should be. For the animal body to maintain homeostasis, cell-cell communication is absolutely essential. There are different strategies that have been evolved where cells are able to communicate with one another. One of them includes the production of electrical impulses. These are usually electrical signals that pass from a cell from one end to another and are then detected by another cell, and they are able to carry the message forward. Electrical impulses are very commonly used by neurons in order to mediate cell-to-cell communication. The production of molecules is also used as a strategy to communicate. These molecules can be inorganic molecules, organic molecules, or more complex molecules like proteins and peptides. They can be secreted where the cell produces the molecules and then secretes it into the extracellular environment. We can also have scenarios where these molecules are actually present on the membrane of the cell. Whether the molecule is secreted or present on the cell membrane, these molecules are usually called as ligands. The ligand can act as a messenger by activating a process called as a signal transduction pathway. The first step of this pathway involves the ligand binding to a protein that is present on the cell called as a receptor. This binding activates the receptor protein, and now we see the process of signal transduction. This is where different proteins are activated in the pathway. And as we can see, one protein activates another protein, and another protein activates other types of proteins. Thus, different proteins get activated. And finally, effector proteins get activated. And these effector proteins are the ones that will generate a cellular response. This cellular response can be in the form of altered metabolism, or changes in gene expression, or changes in cell shape or movement, or all of the above. The ligands, which are extracellular signal molecules, can act as messengers on cells that are nearby or far away. Ligands that are present on membranes are sensed by their target cells through direct cell-to-cell contact. This type of signaling is called as juxtacrine signaling. In this scenario, we are able to observe that the signaling cell has the membrane-bound ligand, while the target cell has the receptor, which is able to bind to that ligand. This usually activates a signaling pathway in the target cell. Juxtacrine signaling is normally observed during the development of the animal, which is when the animal zygote is formed and then undergoes changes to form the multicellular animal. Juxtacrine signaling is also observed in the generation of immune responses, which is used to protect the animal from different types of pathogens. Ligands that are secreted by cells can be sensed by the cell that is making that ligand, or nearby cells, or even cells that are farther away. In the case of autocrine signaling, we have a scenario that the cell that is able to secrete the ligand will also make a receptor protein that can bind to that secreted ligand and allow that cell to respond to the presence of that ligand. In the case of paracrine signaling, the ligand that is secreted by the cells is able to bind to receptors that are present on cells that are nearby or are neighboring cells. In the case of endocrine signaling, the cell that secretes the ligand, which is the endocrine cell, is much farther away than the target cells, which have receptors to bind to the ligand. In such scenarios, the ligand, which is also called as a hormone, is normally carried by blood to these far distances. Synaptic signaling is a special type of paracrine signaling that is usually associated with neurons. The neurons normally secrete chemical molecules called as transmitters at a place called as a synapse, which is between the neuron and the target cell. These neurotransmitters are then sensed by the target cell, which has receptors that will bind to these ligands. In this talk, we will focus on hormones, which are chemical molecules that are secreted into the extracellular fluid of cells and then carried by blood to long distances where the target cells are. They play important roles in homeostasis and can act at a distance from the cell that makes the hormone. There are three main types of hormones. These include peptides, which are short sequences of amino acids, or proteins, which are polypeptides, or different types of polypeptides that associate with one another. Hormones can also be amino acid derivatives, or they can be organic compounds like steroids. The endocrine system comprises of the tissues and organs that are known to secrete hormones. These tissues and organs are ductless and hence secrete the hormones into the extracellular fluid. The hormones are then carried by blood to different parts of the body. The endocrine system is different from exocrine tissues and glands, which secrete their appropriate products into a duct, which is a tube-like structure. When we compare the endocrine and the exocrine systems side by side, we notice that both of them make different types of molecules that are important in physiological processes. Let us focus on the exocrine glands that are known to make secretions like tears, saliva, bile, digestive juices, etc. Let us focus on saliva, which is made by salivary glands that are present in the mouth. The saliva made by the salivary glands are transported to the mouth using the salivary gland ducts. This is different from the endocrine system, where the endocrine tissues and glands do not have any ducts, and hence all the molecules that they produce are directly secreted into the extracellular environment. In order to maintain homeostasis, hormones are involved in feedback loops. Let us take an example where we have a stimulus, which is the change in pH in the duodenum. The duodenum is part of the small intestine, and its pH is normally in the 7 or slightly alkaline range. However, when food from the stomach comes into the duodenum, the pH can drop, resulting in a low pH. This low pH is then sensed by an endocrine cell, which is the S cell present in the duodenum, and results in the secretion of a hormone called as secretin. This hormone then enters the bloodstream and is circulated throughout the body, where ultimately it ends up on target cells which are present in the pancreas. These target cells then release bicarbonate, and this bicarbonate can then neutralize the low pH in the duodenum. This allows the duodenum to restore its pH. Now, once this has occurred, we no longer have a low pH in the duodenum, and hence no more secretin will be made. This is an example of a negative feedback loop, where there is an inhibition of the response by reducing the initial stimulus. Thus, by changing the pH to what it is supposed to be, there is no more secretion of the hormone. While most feedback loops associated with hormones tend to be negative feedback loops, there are instances of positive feedback loops as well. Let us take the example of an infant that is suckling the breast of its mother in order to obtain milk. In this scenario, the suckling is the stimulus, and it will activate the neurosecretory cells present in the mother. This activation of neurosecretory cells results in the production of the hormone oxytocin. The oxytocin is then circulated throughout the body because it enters the bloodstream and ultimately is able to find the smooth muscle cells present in the mammary glands. There it binds to the receptors present on target cells, and the result is the release of milk. Thus, the response in this scenario is the release of milk from the breast that the infant can feed on. However, this response results in the infant suckling more vigorously onto the breast and results in much more milk being released. This is an example of a positive feedback loop. In positive feedback loops, we see that the stimulus is reinforced in order to produce an even greater response by the hormone. With this, we come to the end of our talk, where we learned about homeostasis, ligands that are produced by signaling cells, and receptors that are produced by target cells. We also learned about signal transduction pathways, which happen once a ligand binds to its receptor, as well as hormones, which are proteins or molecules made by endocrine glands. We also learned how endocrine glands are different from exocrine glands, and feedback loops that are observed in maintaining homeostasis.